All right. So welcome back. Hopefully you've had a break and you are a little more receptive to what we are doing. Yes. All right. So let's start with, and just a small request, instead of all of us getting hyper about all this, is as far as the breaks are concerned, the moment a topic is complete, I will give you a break. Even I need a break. All of us need a break, right? So don't the moment it's 11 o'clock and there are desperate messages for a break, it doesn't sound nice. It's not very polite either. So let me finish a particular topic and then ask for a break, right? And it disturbs me, it disturbs the flow, everything, right? Take a break, which has never happened. Yes, so let's be a little more patient, a little more receptive, and let us do the job that we are meant to do, and that is to finish certain topics today. We have to do our learning, right? So let's all be a little patient, a little polite. Just because I cannot see you does not mean that we can write whatever we feel like. That's not a very nice thing to do. Trolling is never nice, isn't it? All right, so let's go on to the types of axoplasmic transport. Now, as far as types of axoplasmic transport is concerned, just make a few changes here, and that is the two types of axoplasmic transport is the fast axoplasmic transport and slow. Instead of anterograde and retrograde, the better way to describe this is fast and slow. Yes. Now, fast can be divided into fast anterograde, and fast retrograde. Yes, fast anterograde and fast retrograde. And slow is always anterograde. Slow is always anterograde, right? So in other words, if I can write anterograde can be fast or slow. Retrograde is always fast. Have you understood? So this is a slightly better way to classify fast and slow. Fast is anterograde and fast is anterograde or retrograde, and slow is always anterograde. Yes. Now, as far as the anterograde transport is concerned, this is 400 millimeters per day. The slow anterograde is in fact 0.5 to 10 millimeters per day, extremely slow. Yes. So otherwise, there is no difference between fast and slow anterograde. Everything else is the same except the speed. Fast anterograde is 400 millimeters per day. Slow anterograde is always 0.5 to 10 millimeters per day. Anterograde transport is towards the plus end of the microtubules. It is towards the plus end of the microtubules. And I've already discussed with you what is the plus end. Plus end is the assembly end. What is the assembly end? Where those rings are added is called the plus end. Where those rings are removed is called the minus end. Right? So this is towards the plus end of the microtubules. Yes. And this is the molecular motors which are used are kinesin and myosin 5. Molecular motors which are used are kinesin and myosin 5. What are the substances which are going to be transported in an anterograde fashion? I've already told you. What is anterograde? Anterograde means away from the cell body. What are the substances which are going to be transported in an anterograde fashion? Whatever is produced by the cell body. So that means neuropeptides. These are produced by the cell body. They will be transported in an anterograde fashion. Then vesicles, new vesicles produced by the cell body. Mitochondria. They can move in an anterograde fashion. What else can move in an anterograde fashion? Reticulum. Lysosomes can move in an anterograde fashion. Right? Lysosomes can move in an anterograde fashion. Remember, what is this axoplasmic transport? This axoplasmic transport is occurring through the axoplasm. There are microtubules, which is like a rail ka patri. It is a track. Moving on the track are gadis. What are the gadis? Gadis are molecular motors. Which are the molecular motors for anterograde transport? Kinesin and myosin 5. What are they going to transport? What is the cargo for them? Cargo can be neuropeptides, reticulum, mitochondria, lysosomes, anything produced by the cell body. That is the cargo which is being transported. Fast can be, like I said, fast can be anterograde, fast can also be retrograde. Retrograde means towards the cell body, towards the cell body. And what is the rate? Fast retrograde is 200 millimeters per day. 200 millimeters per day. This is towards the minus end, towards the minus end. And the molecular motor which is going to be used is dynein. Dynein. And what are the substances which are going to be transported by, which are the substances which are going to be transported by in a retrograde fashion? This will be neurotropic viruses. Neurotropic viruses will be transported in a retrograde fashion. Which are the neurotropic viruses? For example, rabies. Isn't it? What happens in the case of rabies virus? Bite is on a peripheral part. Rabies virus will go in a retrograde fashion towards the central nervous system. So neurotropic viruses will be transported in a retrograde fashion. Then the tetanus toxin. Tetanus toxin will also has to go from the periphery to the central nervous system, periphery to the central nervous system, and that is also with the help of by retrograde axoplasmic transport, retrograde to axoplasmic transport. Then also nerve growth factors. Nerve growth factors can also go in a retrograde fashion towards the cell body. Remember, anterograde is towards the plus end. That means plus end of microtubules are away from the cell body. Yes, and retrograde transport is towards the cell body. That means or towards the minus end. Minus end will be towards the cell body, right? So this is the what is. If you look at this, when I say, somebody was asking me what is the difference between a microtubule and a molecular motor. Microtubules are these hollow tubes which are present inside the axoplasm. These are hollow tubes which are present inside the axoplasm. They have a plus end away from the cell body, minus end over here. Movement towards a plus end is known as the anterograde transport, and movement towards the minus end is known as the movement towards the minus end is known as the retrograde transport. Yes, retrograde transport. Right? This is what is known as your what moves on these microtubules. Microtubules are simply tracks. What is moving on them are the molecular motors. Which are the molecular motors? Kinesin and myosin 5 for anterograde transport, dynein for retrograde transport. This is as far as your axoplasmic transport is concerned. Yes, axoplasmic transport is concerned. Okay. Now, next, let's have a look at what are the different types of. Let's see what are the different types of neurotransmitters. What are the different types of neurotransmitters? Shubhrajit Basak, rabies is retrograde. Yes, retrograde transport is only one type, and that is fast. Anterograde can be fast or slow, but retrograde is only fast, isn't it? Okay. Fast and slow anterograde are absolutely similar except speed. Fast anterograde is 400 millimeters per day. Slow anterograde is 0.5 to 10 millimeters per day. Okay? All right. Just difference is speed. Megha Chaudhary, Rakshanda, just the speed is different. Otherwise, everything else is the same. Same towards the plus end, same substances are transported, same molecular motors, except the speed is different. Fast anterograde is 400 millimeters per day. Anterograde is 0.5 to 10 millimeters per day. Okay? Now let's see what are the neurotransmitters. Yes, neurotransmitters. Now, as far as neurotransmitters is concerned, neurotransmitters are two types. There are small molecule slowly acting, small molecule slowly acting, and small molecule, rapidly acting, and large molecule slowly acting. ओहो, क्या बेटा आप लोग क्या सुनते नहीं हो? क्यों? अच्छा, फास्ट और स्लो में कोई अंतर नहीं है, सिर्फ स्पीड को लेकर अंतर है, बाकी सब कुछ सेम है। सेम सब्सटेंसेस कैन मूव फास्ट, कैन मूव स्लो। गाड़ी दिल्ली से बॉम्बे तेज जा सकती है, स्लो जा सकती है, सेम चीज है। एंटेरोग्रेड कैन बी फास्ट स्लो, सेम सामान दिल्ली से बॉम्बे जा सकता है, चाहे चाहे वो फास्ट जाए या स्लो जाए। डिफरेंस इज ओनली इन टर्म्स ऑफ स्पीड. This is 400, this is 0.5. Same substances can go either fast or slow. Retrograde की डायरेक्शन उल्टी है, वो बॉम्बे से दिल्ली है, उसकी स्पीड 200 मिलीमीटर्स पर डे है। उसके जो सब्सटेंसेस हैं वो है न्यूरोट्रोपिक वायरसेस, टेटनस टॉक्सिन, नर्व ग्रोथ फैक्टर्स। ठीक है, चलिए अब आगे चलिए। दिस इज न्यूरोट्रांसमीटर्स. न्यूरोट्रांसमीटर्स इज two types. There are small molecule rapidly acting, and you've got large molecule slowly acting. Large molecule and slowly acting. Small molecule rapidly acting and large molecule slowly acting. ठीक है? Now, what are these small molecule rapidly acting? They can be divided into 4 classes. There is class 1, and class 1 only has a single neurotransmitter, which is acetylcholine. Class 2 are the amines. And which are the amines over here? Amines are the catecholamines. So you have epinephrine, norepinephrine, dopamine, serotonin, histamine. Right? Epinephrine, norepinephrine, dopamine, serotonin, histamine. These are your class 2. These are small molecule rapidly acting. You've got large molecule and slowly acting, small molecule and rapidly acting neurotransmitters. Class 1, acetylcholine. Class 2 are amines. Epinephrine, norepinephrine, dopamine, serotonin, histamine. Class 3 are the peptides. And which are the peptides? The peptides are glutamate, and aspartate. Glutamate and aspartate, these are excitatory. And there are 2 which are inhibitory, and that is glycine. Sorry, class 3 are what are known as amino acids, not peptides, amino acids. Amino acid, glutamate, aspartate, glycine, and GABA. What is GABA? Gamma aminobutyric acid. Class 4 is a gas. Class 4 is a gas, which is nitric oxide. Right? So these are the different types of neurotransmitters. These are small molecule rapidly acting neurotransmitters. There are 4 classes of small molecule rapidly acting neurotransmitters. Class 1, acetylcholine. Class 2 are the amines, which is epinephrine, norepinephrine, dopamine, serotonin, histamine. Class 3 are the amino acids, which are glutamate, aspartate. Glutamate, aspartate are excitatory. Glycine and GABA are inhibitory. And class 4 is a gas, which is nitric oxide. Now, glutamate is now there was a question which was asked, which is the most important excitatory neurotransmitter in the CNS, and that is glutamate. 75% of the excitation in the 75% of the excitatory effects, excitation in the CNS is due to glutamate, right? This is glutamate. 75% of the excitation is due to in the CNS is due to glutamate. So glutamate, aspartate, glycine, and GABA. Glycine and GABA are always inhibitory. Glutamate and aspartate are excitatory. This was a neat question which was asked. 75% of the excitatory effects in the CNS are due to glutamate. Which are the large molecule and slowly acting neurotransmitters? Now, these can be hypothalamic peptides. Hypothalamic peptides, right? And the hypothalamic peptides are GnRH, gonadotropin releasing hormone, TRH. Now, these are not just hormones, they are also your slowly acting neurotransmitters, GnRH, TRH, right? These thyrotropin releasing hormone, gonadotropin releasing hormone, right? Examples of your hypothalamic peptides, like I said, these are large molecule, slowly acting neurotransmitters. Even pituitary peptides can also act like neurotransmitters. Pituitary peptides can act like neurotransmitters. Yes, for example, growth hormone can act like a neurotransmitter, not just as a hormone, but as a neurotransmitter as well. Gut peptides can also act like neurotransmitters. Gut peptides can also act like neurotransmitters. For example, CCK. Gut hormones can also act like neurotransmitters. Yes, this is CCK, for instance. CCK. Cholecystokinin, not just a hormone, it is also a neurotransmitter. Right? So this is as far as your rapidly acting and slowly acting. What I'm trying to highlight over here is that hypothalamic hormones such as GnRH, TRH, these are peptides secreted from the hypothalamus. They're not just hormones, they can also act like neurotransmitters. Pituitary peptides can act like neurotransmitters. Gut peptides can act like neurotransmitters. But these are all slowly acting. They're large molecule and slowly acting neurotransmitters. This is as far as your the different types of neurotransmitters are concerned. The small molecule rapidly acting divided into 4 classes, class 1, class 2, class 3, and class 4. Class 4 is nitric oxide. This is nitric oxide. NO is nitric oxide. All right. Now, let us now have a look at, when I look at, if you look at the autonomic nervous system. When you look at the autonomic nervous system, you've got sympathetic and parasympathetic, right? Sympathetic and parasympathetic. Now, when I see the sympathetic, this is preganglionic nerve fiber, and this is the postganglionic nerve fiber. Right? Where are the sympathetic ganglia located? The sympathetic ganglia are located. Sympathetic ganglia, these are the sympathetic ganglia, their location is paravertebral. The location is paravertebral. There are paravertebral chain of sympathetic ganglia. Yes, so they are very short. In the sympathetic nervous system, the preganglionic fibers are very short, postganglionic are very long fibers. Now, as far as the neurotransmitters which are going to be secreted in the sympathetic ganglia, neurotransmitters which are secreted in the sympathetic ganglia, here, all autonomic ganglia, the neurotransmitters which are secreted is acetylcholine. Whereas the postganglionic sympathetic nerve fibers will secrete norepinephrine. Norepinephrine. Sympathetic, preganglionic, cholinergic, postganglionic will secrete norepinephrine. This is your postganglionic sympathetic nerve fibers will be secrete norepinephrine, except to sweat glands. Except the postganglionic sympathetic nerve fibers to sweat glands, they secrete acetylcholine, and some renal blood vessels, not all renal blood vessels, but some renal blood vessels, the postganglionic sympathetic nerve fibers secrete dopamine, right? Predominantly, postganglionic sympathetic nerve fibers to renal blood vessels secrete norepinephrine, but some renal blood vessels, the postganglionic sympathetic nerve fibers are dopaminergic. Let's have a look at the parasympathetic. If you see the parasympathetic, the preganglionic parasympathetic nerve fiber is a very long fiber, and postganglionic is extremely short. Yes, that is because if you look at the parasympathetic ganglia are located in or near the organs. They are located near or in the organs. In or near the organs, they innervate. So the postganglionic parasympathetic nerve fibers are extremely short. Now, which are the neurotransmitters which are secreted here? Parasympathetic ganglia, you've got acetylcholine, and postganglionic will also be acetylcholine. So these are very simple things, but important. If you look at the sympathetic and the parasympathetic, postganglionic sympathetic fibers are long fibers. Postganglionic sympathetic majorly secrete norepinephrine, except to the sweat glands, which are cholinergic, and except to see some medial blood vessels, which are dopaminergic. Parasympathetic, on the other hand, the postganglionic fibers are extremely small. Preganglionic, whether it's sympathetic or parasympathetic, cholinergic. Postganglionic parasympathetic are also cholinergic. Now, this is the, we've already done, Abhishek, we've already done the myelination part. We said type B fiber are preganglionic, right? Preganglionic are always type B fibers, which are myelinated. Postganglionic are unmyelinated fibers. We've done that already. Okay? Okay. Joe Napram, the sympathetic ganglia are in the paravertebral chain. Paravertebral chain. Right? The sympathetic ganglia are located paravertebrally. Yes. Absolutely right, Harshita. Preganglionic are myelinated, type B. Postganglionic are type C, which are unmyelinated. Okay. Now, the next thing which I need to discuss with you over here is what is the transmission in autonomic ganglia. Transmission in ganglia. See, up till now we have done that let's see what is the transmission in autonomic ganglia. Now, this is recent advances, and you know that recent advances. Now, normally, up to now, we've done that the autonomic ganglia, acetylcholine, isn't it? Preganglionic fibers, whether they are sympathetic or parasympathetic, the neurotransmitter secreted is acetylcholine. So a little more detail. Now, transmission in autonomic ganglia. When I look at the transmission in autonomic ganglia, there are two types of responses which are going to be seen here. There are fast response and there are slow responses. Right? There is a fast. There is a fast response and there are some slow responses. Fast response and the slow responses. Fast response is fast EPSP. Fast EPSP, this is because of acetylcholine. This is because of acetylcholine. They act on nicotinic receptors. And the response time is in milliseconds. This is known as a fast response, fast EPSP. Transmission in autonomic ganglia, you have two types of responses, fast response and slow responses. Fast response is of one type, which is called a fast EPSP, and that is because of release of acetylcholine acting on nicotinic receptors, and the response time is in milliseconds. Slow responses can be further subdivided into three types. And which are these three types? There is a slow EPSP. Number two, there is a late slow EPSP. And number three, there is a slow IPSP. EPSP is an excitatory response. IPSP is an inhibitory response. Yes, inhibitory response. Now, slow EPSP is because of acetylcholine acting on M1 muscarinic receptors. And the response time is in seconds. Yes. Late slow EPSP is because of GnRH. That GnRH is not just a hormone, it is also a large molecule slowly acting neurotransmitter. So late slow EPSP is because of GnRH, and GnRH will act on peptide receptors. peptide receptors, and the response time is in minutes. Slow IPSP is because of acetylcholine, which acts on M2 muscarinic receptors, and the response time is in seconds. Right? So these are your fast responses and slow responses. Remember, the fast response is only one type, which is a fast EPSP. Slow responses can be slow EPSP, late slow EPSP, and a slow IPSP. In other words, EPSP can be fast, slow, late slow. IPSP is always a slow response. IPSP is because of action of acetylcholine on M2 muscarinic receptors. Right? Next, M2 muscarinic receptors. In terms of the time taken for response, fast EPSP takes the least time. Fast EPSP, then a slow IPSP, then slow EPSP, and then a late slow EPSP. Right? The least time is taken by, so fast EPSP, this is in milliseconds. Slow IPSP takes lesser time than a slow EPSP. Late slow EPSP is in minutes, right? So in terms of time taken for response, will be fast EPSP, slow EPSP, slow IPSP, slow EPSP, and then a late slow EPSP, right? Okay. Now, this is as far as your the transmission in autonomic ganglia. So very important point to understand is autonomic ganglia have which receptors? Receptors in autonomic ganglia. Another important question. Receptors in autonomic ganglia. Receptors in autonomic ganglia. A, does it have nicotinic receptors? Does it have nicotinic receptors? B, does it do they have muscarinic receptors? Muscarinic receptors. C, do they have peptide receptors, or do they have all of the above? Answer is all. You've got all types of receptors: nicotinic, muscarinic, peptide receptors. Right? Now, this also has been asked as a question. Slow IPSP is due to action on which receptor? M2 muscarinic receptor. These are both neat questions which have been asked. All of the above. So you have receptors present in autonomic ganglia. These are nicotinic, muscarinic, peptide, and answer in fact is all of the above. Okay? So this is as far as your transmission in autonomic ganglia is concerned. Let's have a look at the next topic which I'm taking up with you here, and that is, what do you understand by a synapse? Now, when you look at a synapse, right? Synapse is a connection between two neurons or a neuron and a muscle, isn't it? Now, synapses can be classified into two major types. श्याम सुंदर, ये मिनट एंड सेकंड उल्टा लिख दिया क्या? कहाँ लिखा है? नहीं बेटा, ठीक लिखा है. फास्ट ईपीएसपी इज मिली सेकेंड्स, स्लो आईपीएसपी सेकेंड्स में है, स्लो ईपीएसपी भी सेकेंड्स में है, लेट स्लो ईपीएसपी मिनट्स में है. टाइम टेकन फॉर रिस्पॉन्स अगर आप देखें, सबसे ज्यादा लेट स्लो ईपीएसपी लेगा, उससे कम स्लो ईपीएसपी, उससे कम स्लो आईपीएसपी, सबसे कम फास्ट ईपीएसपी. ठीक है? Are you okay with that? Shyam Sundar, I've checked it. It's okay. Shyam Sundar Yadav. All right. Next, let's see. I said synapses. Now, what are the two types of synapses? The two types of synapses could be a chemical synapse, and or an electrical synapse. Now, what is a chemical synapse? Chemical synapse where there is a release of a neurotransmitter. You have a presynaptic neuron. This is a postsynaptic neuron, right? This is a presynaptic neuron, and this is postsynaptic. presynaptic and a postsynaptic. क्या प्रॉब्लम क्या है? Least time taken is by a fast EPSP. Millisecond is the least time taken. Least time taken is a fast EPSP. Maximum time is taken by a late slow EPSP. Maximum time, isn't it? Minute is more than a millisecond. Least time taken is fast EPSP, which takes a millisecond. Late slow EPSP, which takes minutes. संगीता, वाई की बात नहीं है ये सबसे कम फास्ट EPSP, उससे थोड़ा सा ज्यादा स्लो IPSP, उससे ज्यादा स्लो EPSP, राइट? एंड सबसे ज्यादा समय लेट स्लो EPSP. चलिए, chemical synapses, you've got the release of a neurotransmitter here. A neurotransmitter is released over here, and you have on the postsynaptic membrane, receptors are present. There are receptors present on the postsynaptic membrane. This is a chemical synapse. Majority of the synapses are chemical. Most synapses are going to be chemical. Okay? Most synapses are chemical. Electrical synapses, we've already discussed the electrical synapses. Electrical synapse is because of the presence of gap junctions. Gap junctions are due to proteins called connexins. This is an electrical synapse. In electrical synapse, there is a physical continuity, right? This is a gap junction between two neurons, right? Gap junction, which is responsible. So there is a physical continuity which is present. Physical continuity which is present between the cells, right? That is what is known as an electrical synapse. But electrical synapses are very few. Majority of the synapses are going to be chemical. And remember, this is, like I said, chemical and an electrical synapse. Okay? Okay? Now, please remember, a nerve can transmit in both directions. You have a bidirectional transmission in a neuron. It can transmit in both directions. That means if I look at this nerve fiber here, yes, if I give a stimulus here, an action potential which is generated here, this can travel in both directions. A nerve can transmit in both directions. That means there is a bidirectional transmission which is seen in nerve fibers. Bidirectional transmission which will be seen in nerve fibers. Yes, it can travel in both directions. But transmission across a synapse is going to be unidirectional. Transmission across a synapse is unidirectional. Yes, transmission across a synapse is unidirectional. A nerve can transmit in both directions. A nerve can transmit in both directions, but transmission across the synapse is going to be unidirectional. It can only go from preganglionic to postganglionic because then there is a neurotransmitter which is required, right? So this is an important point. A nerve can transmit in both directions. So the action potential which goes towards the cell body will die out. It will not go to the next neuron, right? But across the synapse, the transmission will always be unidirectional. Please understand, electrical synapses, yes, can transmit bidirectionally, but most synapses are chemical. So if the question says transmission across a synapse, your answer should be unidirectional. Transmission in a nerve fiber, we can be bidirectional. Theoretically, can be bidirectional. All right. Okay. Yes, Debashish Nanda, I said across electrical synapses, it's a bidirectional transmission, but most synapses are chemical. So if the question says transmission across a synapse, your answer should be unidirectional, not bidirectional, because electrical synapses are very few. Majority of the synapses are chemical. So majority of transmission across a synapse is unidirectional. But for a nerve, it will be bidirectional. Okay? Two types of synapses, chemical and electrical, but majority are chemical. Electrical are very few, very few. Majority of the synapses are chemical. All right. Okay. Sakshi Patidar, in a spinal motor neuron, the action potential is generated in the initial segment. That was my point last time. Now I'm telling you that if I stimulate an axon somewhere in the middle, an action potential can be generated here. It can transmit in both directions. Yes, but across the synapse, it will be a unidirectional. Okay. In ganglia, we have only acetyl Pachandran. Transmission in autonomic ganglia is your recent advances. Earlier, you thought that there is only nicotinic receptors in autonomic ganglia, but today we know there are nicotinic receptors, muscarinic receptors, peptide receptors, due to which there are different types of responses, which I have elaborated. Okay? All right. Okay. Now, next is, let us now see. Now, synapses can also be described as synapses. There are different ways of classifying. One way was chemical and electrical synapses. Now, synapses can also be described as excitatory or inhibitory, right? Excitatory or inhibitory. Yes, that means excitatory means that in the post-ganglionic neuron, you have the generation of an EPSP. Inhibitory means in the post-synaptic neuron, you have generation of an IPSP, right? So they can be excitatory or inhibitory. Now, let's take on the next topic. And the next topic is, what are the types of inhibition that you see in What are the types of inhibition that you see in the central nervous system? So I'm going to discuss the types of inhibition, right? Types of inhibition which are seen in the central nervous system. What are the types of the inhibitory synapses? So let's see the first one. First one is called a postsynaptic inhibition. What is postsynaptic inhibition? Let's see the types of inhibitory synapses. Postsynaptic inhibition. Now, what is postsynaptic inhibition? Let's see this in detail. In postsynaptic inhibition, you have a neuron A. This is neuron A and this is neuron B. Postsynaptic inhibition, there is a neuron A, there is a neuron B, there is a synaptic connection between the two. Neuron A is known as a presynaptic neuron. This is known as presynaptic. B is postsynaptic. A is presynaptic, B is postsynaptic. Now, A neuron, which is the presynaptic neuron, right? This is a presynaptic neuron. This releases an inhibitory neurotransmitter. An inhibitory neurotransmitter, I've already discussed it with you, which are the two types of inhibitory neurotransmitters, glycine and GABA. They are always inhibitory. The others can be inhibitory, excitatory. The two neurotransmitters which are always inhibitory are glycine and GABA. Yes? So inhibitory neurotransmitter which is released here is usually glycine. And glycine will now cause an IPSP in B. And what is IPSP? Inhibitory postsynaptic potential, which is a hyperpolarizing local potential in the postsynaptic neuron. So in other words, the presynaptic neuron releases an inhibitory neurotransmitter, which is usually glycine. It produces an IPSP in B, inhibits B. This is called a postsynaptic inhibition. This is a postsynaptic inhibition. Right? That means basically your neuron A here is an inhibitory neuron which releases an inhibitory neurotransmitter, and the inhibitory neurotransmitter is usually glycine, producing an IPSP. This is known as postsynaptic inhibition. The second type of inhibition that you see is what is known as a presynaptic inhibition. This is known as a presynaptic inhibition. right? This is known as presynaptic inhibition. Now, what happens in the presynaptic inhibition is, what happens in the presynaptic inhibition? Here you have a neuron A. You have a neuron A, which forms a synapse with neuron B. Neuron A is presynaptic, neuron B is postsynaptic. So normally what happens in a presynaptic inhibition is neuron A, which is a presynaptic neuron, excites neuron B, right? It excites neuron B. It releases an excitatory neurotransmitter and excites neuron B. A is presynaptic, B is postsynaptic, right? So A excites B. But there is an inhibitory neuron, right? Inhibitory neuron. This is an inhibitory neuron. This is a GABA secreting neuron. This is neuron C. It releases an inhibitory neurotransmitter, which is GABA, right? It releases an inhibitory neurotransmitter, which is GABA. And this neuron C, it forms an axonal synapse. See, anatomically, synapses can be axodendritic, axosomatic, axonal. So here you see an axonal synapse, right? Let's see this once again. What happened in a postsynaptic inhibition was very straightforward. Neuron A, which is the presynaptic neuron, releases an inhibitory neurotransmitter, which is glycine, which produces an IPSP in B and inhibits B. B is postsynaptic. So A releases an inhibitory neurotransmitter, inhibits B. This was postsynaptic inhibition. In presynaptic inhibition, neuron A excites neuron B, but there is a neuron C which forms an axonal synapse with neuron A. A is presynaptic, B is postsynaptic. Normally A excites B, but C, which is a GABA secreting neuron, now forms an axonal synapse with the presynaptic neuron. Now, there are GABA receptors. There are GABA receptors on the presynaptic neuron. GABA receptors on the presynaptic neuron. There are GABA receptors on the presynaptic neuron. And these GABA receptors are of two types. And what are these two types of GABA receptors? These are ionotropic receptors, ionotropic and metabotropic receptors. Ionotropic and metabotropic. What are metabotropic? Metabotropic are G protein coupled receptors. Ionotropic receptors are GABA A, and GABA C. Metabotropic receptors are GABA B. Right? So these are the three types of receptors: GABA A, GABA C, GABA B, and GABA C. Ionotropic, that means they are related to, they cause the opening of ion channels. Metabotropic, G protein coupled receptors. Right? Now, GABA A and GABA C receptors, they increase the chloride conductance. And we have already discussed this, that if there is increase in chloride conductance, that means they basically cause opening of chloride channels. If there is increased opening of chloride channels, I told you last time that if a channel is open, that particular ion will come from higher to lower concentration. So it comes from outside to inside. There is an influx of chloride. And if there is an influx of chloride, that means what will happen to the potential of the neuron? Potential of the cell will become more negative. What is a more negative potential known as? Hyperpolarization, right? So this leads to increase in chloride conductance will lead to a hyperpolarization. There is a hyperpolarization of the presynaptic neuron, hyperpolarization of the presynaptic membrane. Yes? Hyperpolarization of the presynaptic membrane. Let's look at GABA B. GABA B increases the potassium conductance. And again, we have discussed this. If there is increase in potassium conductance, that means there is loss of positive ion, and loss of positive ion means, again, it will lead to a hyperpolarization. There is a hyperpolarization of the presynaptic membrane. If there is either way, whether it is GABA A, whether it is GABA C, whether it is GABA B, ultimately GABA is causing hyperpolarization of the presynaptic membrane. Now, if I look at the diagram, if the presynaptic membrane is hyperpolarized, this membrane is hyperpolarized. Presynaptic membrane means membrane of neuron A. Now, this is hyperpolarized. So that when an excitatory impulse reaches this presynaptic membrane is hyperpolarized because of GABA. Yes? And now, remember, presynaptic membrane normally secretes an excitatory neurotransmitter. So what will happen to the secretion of this excitatory neurotransmitter? This will now reduce. Reduction in the release of an excitatory neurotransmitter is also a type of inhibition, indirectly. This is what is known as your presynaptic inhibition. Have you understood? Here, presynaptic membrane is getting hyperpolarized because of GABA, so that when a normal action potential reaches this membrane, normally the presynaptic membrane releases an excitatory neurotransmitter. Now the release of that excitatory neurotransmitter is reduced. Reduction in the release of the excitatory neurotransmitter is also a type of inhibition, which is known as a presynaptic inhibition. See, postsynaptic inhibition was direct. Release of an inhibitory neurotransmitter inhibits a postsynaptic. Here, what has happened is the GABA has caused a hyperpolarization of the presynaptic membrane, decrease the release of excitatory neurotransmitter. And this is exactly an Ames question which was asked, and that was presynaptic inhibition is due to A. Is it due to hyperpolarization of the presynaptic membrane? Is it due to a hyperpolarization of the presynaptic membrane? Or B, is it due to an inhibition of release of excitatory neurotransmitter? Inhibition of release of excitatory neurotransmitter by the presynaptic membrane. Right? Inhibition of release of the excitatory neurotransmitter by the presynaptic membrane. Right? Bothello, Mark Richard, chloride influx or efflux? Basics, beta, please do not forget your basics. An ion can only move from higher to lower concentration. Chloride is higher outside the cell. So there can only be influx of chloride. You cannot get an efflux of chloride through a channel. Now, I've asked you this question, this was an Ames question, and that is it due to hyperpolarization of the presynaptic membrane or inhibition of release of excitatory neurotransmitter? Answer is hyperpolarization. Please remember, hyperpolarization of the presynaptic membrane leads to not an inhibition of release of excitatory neurotransmitter, it leads to a decrease in the release of excitatory. There can never be a complete inhibition. It can only be a decrease in the rate of potassium, a decrease in the rate of decrease in the release of the excitatory neurotransmitter. So my better answer is A. Right? I will not see. Another reason why I prefer A over B is because there is never an inhibition. It is only a decrease in the release of excitatory neurotransmitter. Right? See, this was an Ames question. You did not have an option of both. You had A as your correct answer. Okay. Now, increase in potassium conductance means increase in potassium efflux. More potassium will leave the cell, so resulting in a hyperpolarization. Now, one particular area where you see this presynaptic inhibition is in the motor pathway. Now, if you look at the motor pathway, the tract which carries impulses from the cortex to the spinal cord, that is known as the lateral corticospinal tract. Right? This is the motor pathway here. What is the motor pathway? Like I said, Let's see the motor pathway. This is the lateral corticospinal tract, and the lateral spinal tract, which is also known as the pyramidal tract. Now, this is the presynaptic neuron here. It forms a synapse with the A-alpha motor neuron. We've discussed this last time as well, right? This is the A-alpha motor neuron, or also known as the spinal motor neuron. Yes, we discussed this briefly last time. This is the motor pathway. Yes. Now, presynaptic here is the pyramidal tract neuron, which is also called the upper motor neuron, and the A-alpha motor neuron, or the spinal motor neuron, is also known as the lower motor neuron. So normally what happens is the pyramidal tract neuron releases an excitatory neurotransmitter, which is glutamate. And glutamate will now, it is excitatory. I've told you 70%, 75% of the excitation of the central nervous system is caused by glutamate. This glutamate will now excite the lower motor neuron. It excites the lower motor neuron, and so impulses from the cortex, pyramidal tract, release of glutamate by the pyramidal tract neuron, excites the spinal motor neuron. Impulses will now go towards the muscles, right? Impulses will go towards the muscles, and a transmission across the neuromuscular junction and muscle contraction. This is your motor pathway, right? Now, present within the spinal cord, there is a small GABA secreting neuron. This secretes GABA. This is a GABA secreting neuron. This is present within the spinal cord. This GABA now causes a hyperpolarized synaptic membrane. And which is the presynaptic membrane? Membrane of the pyramidal tract neuron, membrane of the upper motor neuron. This is now hyperpolarized. So when this is hyperpolarized, right? Hyperpolar, baby. When this is hyperpolarized, now what happens is when an excitatory impulse reaches the end of the upper motor neuron. Remember, there was release of glutamate here. Now, because the membrane is hyperpolarized, the release of glutamate now reduces. Yes, release of glutamate now reduces. This is a presynaptic inhibition of the motor pathway. GABA causes hyperpolarization to presynaptic membrane, reduces the release of the excitatory neurotransmitter. What is the advantage of a presynaptic inhibition? Advantage of a presynaptic inhibition is it prevents an excessive excitation. This prevents an excessive excitation. It prevents an excessive excitation of the lower motor neuron. It prevents an excessive excitation of the lower motor neuron, and that in turn leads to a precise and a well-controlled motor movement. This results in a precise and a well-controlled motor movement. This is the advantage of a presynaptic inhibition of the motor pathway. It prevents an excessive excitation of the lower motor neuron. See, motor movements are extremely precise, controlled. So, of course, there is the role of the cortex, there is a role of the cerebellum, there is a role of the basal ganglia, but even at the level of the spinal cord, there is control which is present. One of them is this presynaptic inhibition. Presynaptic inhibition, right? This is as far as your, like I said, advantage of a presynaptic inhibition of the motor pathway. It prevents an excessive excitation of the lower motor neuron, and that it results in a precise and a well-controlled motor movement. Okay? Now let's have a look at what happens in the case of tetanus toxin. Please, if your apps are, there is a hang, refresh your feed. I'm also watching the complete class on my own phone. There is no hang in my phone. I'm looking at the class just like you are. So there is no hang. If there is a hang, please refresh your feed. Now let us see. Now, Prizal Sharma, please remember, majority of post-synaptic inhibition majorly is produced by glycine. Majority of the presynaptic inhibition is produced by GABA. That is why we talk about this. Have you understood? Right. This If your app is crashing, please refresh your feed. Okay, now let's see this here. Now what happens in the case of tetanus? Now in tetanus toxin, let's see what happens in case of tetanus. Now tetanus will inhibit, tetanus toxin inhibits the release of GABA, right? It inhibits the release it inhibits the presynaptic inhibition. Yes, tetanus toxin will act on the spinal cord. It inhibits the release of GABA. So if it inhibits the release of GABA, that means it inhibits the presynaptic inhibition. Two negatives will make a positive. If two negatives make a positive, what will happen? Two negatives will make a positive. There is going to be an excessive release of glutamate. And the type of paralysis which is produced in tetanus is known as spastic paralysis. Yes, this is your the tetanus. The type of paralysis which is produced is going to be a spastic paralysis. The type of paralysis which is produced is going to be spastic, right? Botulinum toxin acts at the neuromuscular junction, which we've done last time. It produces a flaccid paralysis, but tetanus toxin acts on the spinal cord. It inhibits the presynaptic inhibition and produces a spastic paralysis. Okay? This is as far as your when you look at the बेटा वो सब बोल रहा है ऐप क्रैश कर रहा है, बिल्कुल नहीं कर रहा है, मेरे को तो दिखाई दे रहा है, नहीं मेरे को तो ये क्यों, हाँ तो ये क्यों, हाँ इसको आप, हाँ, मैं भी देख रही हूँ, कोई नहीं, कोई इशू नहीं है. All right, so let's have a look at the motor pathway. Let's see this. You've got the pyramidal tract neuron, which releases an excitatory neurotransmitter to stimulate the lower motor neuron. There is a GABA secreting neuron, which is present in the spinal cord. It causes a hyperpolarization of the upper motor neuron, which decreases the release of the excitatory neurotransmitter. What is the advantage of a presynaptic inhibition? It prevents an excessive excitation of the lower motor neuron. This leads to a precise and motor movement. Now, what is the action of tetanus toxin? Tetanus toxin acts on this presynaptic inhibition, and it inhibits the presynaptic inhibition. What is inhibition of a presynaptic inhibition? Inhibition of an inhibitory mechanism. Two negatives will make a positive. So what happens is, now when tetanus inhibits release of GABA, it now causes, there is an excessive release of glutamate. Yes. So tetanus toxin, what is the effect of tetanus toxin? It inhibits release of GABA. It inhibits the presynaptic inhibition. Right? It inhibits the presynaptic inhibition. It inhibits the release of GABA. And what is GABA? inhibitory neurotransmitter, inhibits release of GABA. So two negatives will make a positive. Inhibition of release of an inhibitory neurotransmitter, that means now there is going to be an excessive release of glutamate. There is an excessive release of glutamate, right? There is an excessive release of glutamate by the upper motor neuron. There is an excessive release of glutamate by the upper motor neuron, and that in turn now results in what is known as a spastic paralysis. Spastic paralysis, yes? This results in a spastic paralysis. We've done botulinum toxin. Botulinum toxin acts at neuromuscular junction, produces a flaccid paralysis. Tetanus toxin acts on the spinal cord, inhibits a presynaptic inhibition. There is excessive release of glutamate by the upper motor neuron, resulting in a spastic paralysis. Right? Spastic paralysis. So we've done two types of inhibition. We've done postsynaptic inhibition, presynaptic inhibition. Let's have a look at the third type of inhibition, which is called the feedback inhibition. The third type of inhibition is known as a feedback inhibition. Feedback inhibition. Now, feedback inhibition is also seen in the motor pathway. Now, again, let's discuss motor pathway. This is the pyramidal tract neuron. Yes. This is the pyramidal tract neuron. Yes, this is your upper motor neuron. Pyramidal tract neuron or the upper motor neuron, it synapses with the spinal motor neuron, which goes to the muscles. This is the lower motor neuron. And the upper motor neuron excites lower motor neuron. There is release of glutamate, as I have already discussed. Right? There is glutamate, as I've already discussed. Now, this lower motor neuron, it gives rise to a small collateral. And this synapses with an inhibitory neuron which is present within the anterior horn of the spinal cord, and this is known as the Renshaw cell. Renshaw cell is an inhibitory neuron which is present in the anterior horn of the spinal cord. It is present in the anterior horn of the spinal cord, and this Renshaw cell now forms a synapse with the same alpha motor neuron. Remember, this is a small inhibitory neuron present in the anterior horn of the spinal cord. What is this Renshaw cell? This is a small inhibitory neuron present in. This is a small inhibitory neuron which is present in the anterior horn of the spinal cord. Present in the anterior horn of the spinal cord. Right? And this in turn now forms a connection with the same alpha motor neuron. So basically what happens is, when the upper motor neuron, it sends an excitatory impulse, there is release of glutamate, which excites the lower motor neuron. Impulses will go towards a muscle which is responsible for muscle contraction. But some impulses will also go to the Renshaw cell. They will excite the Renshaw cell, but Renshaw cell will release an inhibitory neurotransmitter, and it inhibits the same alpha motor neuron. It is sort of a negative feedback. Have you understood? And I described it in the first chapter. I told you negative feedback is always a good thing. Yes? What is the advantage of this feedback inhibition? Advantage is, again, it prevents an excessive excitation. Right? It prevents an excessive excitation of the lower motor neuron. It prevents an excessive excitation of the lower motor neuron, and that in turn leads to, it prevents an excessive excitation of the lower motor neuron, which in turn leads to a precise and a well-controlled motor movement, right? Precise and a well-controlled motor movement, right? This is the advantage. It prevents excessive excitation of the lower motor neuron, which in turn leads to a precise and a well-controlled motor movement. So this is what is known as a feedback inhibition, right? We've done 3 types of inhibition. I can revise it for you once again. The first one was what is called as postsynaptic inhibition, where the presynaptic neuron releases an inhibitory neurotransmitter, which is usually glycine, producing an IPSP. IPSP is a hyperpolarizing local potential in B, which inhibits B. In presynaptic inhibition, there is a GABA secreting neuron which forms an presynaptic. Release of GABA causes a hyperpolarization of the presynaptic neuron and decreases the release of excitatory neurotransmitter from the presynaptic neuron. Decrease in the release of excitatory neurotransmitter is also a type of inhibition. I asked you an Ames question here. What is it due to? It is due to hyperpolarization of the presynaptic membrane or inhibition of release of excitatory neurotransmitter. Better answer is A. Then I discussed with you what happens in tetanus toxin. Tetanus toxin will inhibit the presynaptic inhibition. It inhibits the release of inhibitory neurotransmitter, which in turn leads to an excessive release of glutamate by the upper motor neuron, causing a spastic paralysis. Let's see feedback inhibition. Feedback inhibition is Renshaw cell inhibition. What is Renshaw cell? It is a small inhibitory neuron present in the anterior horn of spinal cord. As the presynaptic neuron excites the postsynaptic, as the upper motor neuron excites the lower motor neuron also stimulates the Renshaw cell. But Renshaw cell, once it gets stimulated, will release an inhibitory neurotransmitter, causing a partial inhibition of the lower motor neuron. Advantage: prevents an excessive excitation of the lower motor neuron. Now, let's see what is the 4th type of, there are 5 types of inhibition. We've done 3. Let's go on to the 4th type of inhibition. 4th type is called what is known as the feed-forward inhibition, right? This is known as a feed-forward inhibition. Now, what happens in the case of feed-forward inhibition? Let's see that. Now, feed-forward inhibition is, here, neuron A, right? There are 3 neurons here. Neuron A excites neuron B. Right? Neuron A excites neuron B, which in turn inhibits neuron C. Right? A excites B, but B inhibits C. Right? Here, that means basically neuron B is an inhibitory neurotransmitter. It releases an inhibitory neurotransmitter, which now inhibits B. A excites B, but B inhibits C. This is feed. B is an inhibitory neuron, releasing an inhibitory neurotransmitter. One example where you find feed-forward inhibition is in the cerebellum. Let's see what happens in the cerebellum. In cerebellum, the granule cell excites the basket cell. Granule cell excites a basket cell, but basket cell inhibits the Purkinje cell. Right? Granule cell excites a basket cell, but basket cell inhibits the Purkinje cell. Another example which is present in the cerebellum. So A excites B, but B is inhibiting C. That is what is known as feed-forward inhibition. Another example which you see in the cerebellum is granule cell will excite the stellate cell. We'll be doing this in detail when I do cerebellum with you. This will excite the stellate cell, but stellate cell will inhibit the Purkinje cell. This is what is known as a feed-forward inhibition. A excites B, but B inhibits C. Right? So this is as far as your feed-forward inhibition is concerned. Right? Okay. Then the 5th type of inhibition that you see is what is known as a lateral inhibition. Lateral inhibition. Yes, lateral inhibition. Now, what is a lateral inhibition? Right? Lateral inhibition. Now, lateral inhibition is, for instance, you see lateral inhibition, for example, in the retina. Right? you see a lateral inhibition in the retina, isn't it? Now, for example, if this is a rod which is getting excited, light falls on this, it excites this rod here, right? Now, via the horizontal cells, yes, this rod will inhibit these set of rods which are getting excited. They will inhibit the neighboring rods. That is known as a lateral inhibition, right? When light falls on this group of rods here, via the horizontal cells, they inhibit the neighboring rods, right? They inhibit the neighboring rods. That is known as neighboring rods, and that is known as a lateral inhibition. What is the advantage of this lateral inhibition? Lateral inhibition will increase the acuity. Yes, the rods which are getting excited, they inhibit the neighboring rods via the horizontal cells. So if surrounding rods are inhibited, that sort of increases the acuity, gives you a sharp image with a well-defined margins, right? This gives an image with a well-defined margin. With well-defined margins. That is what is known as lateral inhibition, with well-defined margins. This is what is known as increases the lateral inhibition. You can also see this in the sensory system, yes? Now, for instance, you've got, let's say, there are two receptors. You've heard of two-point discrimination. Two-point discrimination. What is two-point discrimination? Two-point discrimination is the ability to detect 2 points as two separate points. That is called two-point discrimination. Yes, you excite 2 points on the skin simultaneously. Your ability to feel them, to detect them as two separate points is known as two-point discrimination. Two-point discrimination is also based on lateral inhibition. This phenomena is also based on lateral inhibition. Yes, 2 points on the skin are excited simultaneously. These receptors are getting excited here. And these receptors which are getting excited, they inhibit the neighboring receptors. They send inhibitory impulses to the neighboring receptors. So you're able to detect these 2 points as two separate points. This is based on lateral inhibition. This was two-point discrimination is based on which physiological phenomenon? This is based on lateral inhibition. Lateral inhibition increases the sensory acuity here. Here it is increasing the sensory acuity, right? This is what is known as the lateral inhibition. Yes. Now, just to complete this, you know what is minimum separable. What is minimum separable? What is minimum separable? Minimum separable is the minimum distance. This is the minimum distance between 2 points to be able to perceive them. Minimum distance between 2 points to be able to perceive them as two separate points. To be able to perceive them. To be able to perceive them as two separate points. That is known as two separate points. That is known as minimum separable. Minimum separable. 2-point discrimination. Minimum separable, minimum distance between 2 points to be able to perceive them as two separate points. This is on the fingertips. Minimum separable on the fingertips. This is as little as 2 millimeters. 2 millimeters. On the back, the minimum separable is 65 millimeters. 65 millimeters. That means on the back, if I excite 2 points, which are a distance of, let's say, 50 millimeters, perceive those 2 points as 1 point. To be able to perceive them as two separate points, the distance on the back should be between two pinpoint stimuli, has to be at least 65 millimeters. That is what is meant by minimum separable. minimum separable. Okay? So this is as far as your 2-point discrimination is concerned, right? So again, let's see your 5 types of inhibition. The first 2 are very simple: postsynaptic, presynaptic. Postsynaptic, direct. Presynaptic neuron releases a inhibitory neurotransmitter and inhibits the postsynaptic neuron. Presynaptic inhibition, GABA secreting neuron, which causes hyperpolarization of the presynaptic membrane, decreases the release of excitatory neurotransmitter. What is feedback inhibition? This is also known as the Renshaw cell inhibition. Yes, there is this Renshaw cell is a small neuron present in the anterior horn of the spinal cord, has been asked as a question, and this causes a feedback inhibition of the same alpha motor neuron. But example, like I said, what is the advantage? Precise, well-controlled motor movement. Feed-forward inhibition, typically seen in the cerebellum. A excites B, but B inhibits C. Granule cell excites a basket cell, which inhibits a Purkinje cell. What is lateral inhibition? Typical example seen in the retina. A group of rods which are getting excited, they now inhibit the neighboring rods via the horizontal cells. This increases the acuity, gives images with well-defined margins. 2-point discrimination is based on lateral inhibition. This is based on the phenomena of lateral inhibition. It increases the sensory acuity. Minimum separable between 2 points to be at the fingertips 2 millimeters, at the back 65 millimeters. Okay, so this is as far as your 5 types of inhibition are concerned: postsynaptic, presynaptic, feedback, feed-forward, lateral inhibition. All right. Let's go on to the next topic. The next, which I am going to discuss with you now, is the sensory system. Let's go on to the sensory system. Okay, let's see sensory. Manendra Prasad, feed-forward inhibition, I will discuss it with you in detail when I do cerebellum. Don't worry, we'll discuss it. Okay? All right. Now, as far as the sensory system is concerned, let's see. Now, the first thing as far as the sensory system is concerned, the first thing is receptor. Please speak slow. Lily Chanu, what do you mean slow? Do you want me to reduce my volume or reduce the speed? Lily Chanu? Lily Chanu, what do you want, beta? Reduce the volume, reduce the speed. Speed, okay. I'll try my best, Lily Chanu. Neil Harish, I'll try my best. Okay. Receptor. But at the same time, Lily Chanu and Harish, I am writing everything that I am speaking. So you should not have everything I'm writing down. Just because so that your notes can be proper. So even if you have a problem in understanding my English, I'm writing everything down, isn't it? All right. Now, what is a receptor? Now, what is the definition of a receptor? Okay. Fine. Just stop typing. Stop typing, please. Bus. Shanti rakho, please. Receptor. Now, what is a receptor? Receptor is a biological transducer. Receptor is a biological transducer. What is a transducer? What is a transducer? Transducer is anything which converts one form of energy to a different form of energy. That is what is known as a transducer, biological transducer. It converts one form of energy to another form of energy, isn't it? So a receptor is a biological transducer, right? Biological transducer. See, after all, what are touch receptors? Touch receptors will convert touch, mechanical sensation into action potentials. Rods and cones in the retina are converting light energy to action potentials. Right? The hair cells of the cochlea, they convert sound energy to action potentials. So what is a transducer? Transducer converts one form of energy to another form of energy. Converts one form of energy to another form of energy. Right? It converts one form of energy into another form of energy. Yes? And so receptors are nothing but biological transducers. Now, we've already discussed what is a sub-threshold stimulus, what is a threshold stimulus. Sub-threshold stimuli will produce local potentials. What are threshold and supra-threshold stimuli? They will produce action potentials. We discussed this last time, right? Now, what do you understand? The next is, what do you understand by an adequate stimulus? What is an adequate stimulus? What is an adequate stimulus? Yes? Now, this is again a question which was an Ames question. What is an adequate stimulus? Now, adequate stimulus is, now for example, a very simple example, rods and cones in the retina are responding to light. They will also respond to pressure. That means if I give a pressure on the eyeball, I get flashes of light. But the rods and cones are more sensitive to light or pressure. They are more sensitive to light. Pressure has to be intense to be able to stimulate the rods and cones. Yes? So what is the adequate stimulus for rods and cones? This is going to be light. Yes? What is adequate stimulus? Let's see what is adequate stimulus. Adequate stimulus is the stimulus. Adequate stimulus is the stimulus to which a receptor. Adequate stimulus is a stimulus to which a receptor is most sensitive, or to which a receptor responds at, low energy levels. What is adequate stimulus? Adequate stimulus is the stimulus to which a receptor is most sensitive, or to which a receptor responds at low energy levels. For example, like I told you, adequate stimulus for rods and cones. Adequate stimulus for rods and cones is light and not pressure. Remember, rods and cones can respond to pressure, but the pressure has to be intense to be able to stimulate the light rods and cones. So adequate stimulus for rods and cones is light and not pressure, right? So threshold stimulus and sub-threshold, supra-threshold, those are that is, adequate stimulus is not threshold stimulus. Adequate stimulus is the stimulus to which a receptor is most sensitive, or to which a receptor responds at low energy levels. Yes, this is adequate stimulus. Now, then, the next important point. What do you understand by sensory coding? What is sensory coding? Yes? What is sensory coding? Yes? Now, sensory coding, a receptor codes for. Receptor codes for the 4 attributes of a stimulus. A receptor codes for 4 attributes of a stimulus. Receptor codes for 4 attributes of a stimulus. And which are the 4 attributes of a stimulus? This is site or location. Number 2, modality. Number 3, modality. Number 1, site or location, modality. Number 3, intensity. And number 4 is duration. These are the 4 attributes of a stimulus. Basically, this is what is meant by sensory coding. What are the 4 attributes? Again, this has been asked as a question. What are the 4 attributes of a stimulus? That means, basically, this means that the receptor will code for, or receptor gives me information about the 4 important points of a stimulus. And which are these 4 important points? The 4 important points is it will tell me, a receptor will tell me what is the exact site or location of a stimulus, what is the modality. When I say modality, that means, for example, is it fine touch? Is it crude touch? If it is fine touch, has it been done by a cotton wool or by a silk cloth or by a feather? So that is modality, intensity, and duration. So these are your 4 attributes of a stimulus. A good receptor will tell me that a good receptor will tell me what is the exact site or location of the stimulus, what is the modality, what is the intensity, and what is the duration. This is known as sensory coding. Now, the next important point is, what do you understand by adaptation of a receptor? What is adaptation of a receptor? What is adaptation? Adaptation means that the stimulus is present. Adaptation of a receptor, stimulus is present, but the response decreases over a period of time. Response decreases over a period of time. That is known as adaptation. What is adaptation? Adaptation is that stimulus is present, but response has reduced over a period of time. That is known as adaptation. Okay? Stimulus is present, but the response reduces over a period of time. For example, a simple thing, you walk into a room, yes, there are some dirty socks, smelly socks. The smell is overpowering when you enter a room. But after some time, you continue, if you have to stay in that room due to whatever reason, after some time, your olfactory receptors have adapted. They stop responding. Have you understood? Right? You are comfortable with that smell after a little while. That is what is meant by adaptation. Stimulus is present, but the response has reduced and may even disappear, isn't it? That is what is known as adaptation. Now, depending upon the time taken for adaptation, there are 2 types of receptors. There are 2 types of receptors. There are, you have rapidly adapting receptors. There are rapidly adapting receptors. There are rapidly adapting receptors, and there are slowly adapting receptors. Yes, depending upon the time taken for adaptation, you have the rapidly adapting receptors and slowly adapting receptors, right? So the receptors can be divided into 2 types. You've got rapidly adapting receptors and slowly adapting receptors. Now, rapidly adapting receptors, let's see, which are the rapidly adapting receptors? These are also known as phasic receptors. These are also known as phasic receptors, right? Whereas the slowly adapting receptors are also known as tonic receptors. Tonic receptors. Phasic receptors are these are encapsulated nerve endings. These are encapsulated nerve endings. These are encapsulated nerve endings. Slowly adaptive receptors, these are expanded or free nerve endings. These are expanded or free nerve endings. Yes, encapsulated nerve endings, these are expanded or free nerve endings. Rapidly adapting receptors, they can detect rate of change of stimulus. Rate of change of stimulus. When I say rate of change, I've already told you when you use the term rate, that means per unit time. Yes? Supposing you have an increasing strength of stimulus or a decreasing strength of stimulus. I've understood. Rate of change means when the stimulus strength is changing with time. It could be an increasing strength of stimulus or a decreasing strength of stimulus. This will be detected by the phasic receptors. Whenever there is a rate of change, strength of stimulus changes with time, right? Strength of stimulus changes with time. Whereas this can detect a steady stimulus, right? This can detect a steady stimulus. What is a steady stimulus? Here the stimulus strength is steady over a period of time. This can be detected by a tonic receptor, right? Rate of change of stimulus, this is by a phasic receptor. The steady stimulus will be by a tonic receptor, right? This is what is known as see, a very simple kind of a, let me try and explain this to you. Supposing I give a steady stimulus. What is a steady stimulus? Stimulus means when the stimulus strength is constant over a period of time, right? Stimulus strength is constant over a period of time. This is a steady stimulus. Now, what is going to be the response of a phasic receptor? Response of a phasic receptor is initially it shows an action potential, but because it is rapidly adapting, it stops responding. Have you got it? No action potentials in the phasic receptor. So phasic receptor is rapidly adapting, so it stops responding, right? It stops responding. But if I give a steady stimulus to a tonic receptor, what is the response of a tonic receptor? Tonic receptor will show an action potential. It will continue to show an action potential. If it continues to show an action potential, that means it continues to respond because it is slowly adapting. So which receptor has been able to detect a steady stimulus? That is going to be a tonic receptor, not a phasic receptor. Phasic receptor stopped responding. Have you understood? So steady stimulus will be detected by a tonic receptor, not by a phasic. Phasic will show response initially, but then stops responding because it is rapidly adapting. Adaptation means stimulus is present, but the response has decreased over a period of time. Right? Okay. Now, what are the examples of rapidly adapting receptors? Examples of rapidly adapting receptors, let's say Pacinian corpuscle. Pacinian corpuscle is your rapidly adapting receptors. Pacinian corpuscle is a rapidly adapting receptor. The nerve fiber which is associated with Pacinian corpuscle is A beta. So what happens? What does a Pacinian corpuscle look like? Pacinian corpuscle, now if this is the A beta nerve fiber, A beta, if you remember, A beta nerve fiber is a myelinated nerve fiber, right? This is a myelinated nerve fiber. This is A beta, right? The tip of this A beta nerve fiber is encapsulated. I just told you the rapidly adapting receptors are encapsulated. There are multiple layers of capsule here. Yes, there are multiple layers of capsule. This is encapsulated. Yes, this lies under the skin. Yes, this lies under the skin. This is your Pacinian corpuscle. A beta nerve fiber is a nerve which is associated with Pacinian corpuscle. The tip of this A beta nerve fiber is encapsulated. There are multiple layers of capsule. This is a rapidly adapting touch receptor. Right? What is this? Pacinian corpuscle is a very rapidly adapting touch receptor. This is a very rapidly adapting touch receptor. Very rapidly adapting touch receptor. Precedent corpuscle is specific for fast vibration. This is specific, this is not meant for touch, this is meant for fast vibration. Specific for fast vibration up to 800 cycles per second. Specific for fast vibration up to 800 cycles per second. And if you remember your sensory system examination in the first year, if you remember testing for vibration, how did we test for vibration? We took tuning forks. Tuning forks were taken and they were placed on bony prominences. What was the frequency of the tuning fork? The frequency was 128 hertz. 256 and 512 hertz. Tuning forks with 256 and 512 hertz are used for tests of hearing. 128 hertz was used for testing vibration. How did we test for vibration? We took these tuning forks, we placed them on bony prominences. Why did we place them on bony prominences? Precedent corpuscles, which are receptors for fast vibration, these are present in joint capsules. Precedent corpuscles are present in joint capsules. So they helped to detect vibration on bony prominences. They are present in joint capsules. They are also have a deep location. Precedent corpuscles are located deep in muscles, fasciae. So these receptors, since they have a deep location, they can also be used for detection of deep pressure. I've already told you what is deep pressure, poking, right? So these are receptors for fast vibration up to 800 cycles per second, and also precedent corpuscles will detect deep pressure. Deep pressure, we've already discussed that. What is deep pressure? That is poking. Because remember, precedent corpuscles have a deep location. You do not have precedent corpuscles just under the skin. You have precedent corpuscles in joint capsules, therefore meant for detection of vibration, and also deep in the skin, muscles, fasciae, they can detect deep pressure, such as poking. So this is your precedent corpuscles. Yes? Let's have a look at another rapidly adapting receptor, which is known as the Meissner's corpuscle. Another rapidly adapting receptor is the Meissner's corpuscle. Meissner's corpuscle the nerve fiber which is associated with the Meissner's corpuscle is again A beta, right? Precedent corpuscles located in joint capsules and deep in muscles and fasciae. Precedent corpuscles located for detection of specific for fast vibration up to 800 cycles per second, and also for detecting deep pressure. Okay? All right. Shanu Soni, these are not meant for touch. They are rapidly adapting touch receptors, but they are not meant for touch. They are meant for fast vibration up to 800 cycles per second. Meissner's corpuscles. Meissner's corpuscles, the nerve fiber associated with Meissner's corpuscle is A beta. Now, Meissner's corpuscles, they are meant for fine touch. They are meant for fine and well-localized touch. Fine, well-localized touch. They are meant for slow vibration. Slow vibration up to 80 cycles per second. Cycles per second can also be written as hertz, means the same thing. Cycles per second or hertz. Right? Slow vibration up to 80 cycles per second. Meissner's corpuscles are also meant for Meissner's corpuscle. These are also meant for texture. Texture. You know, when you are trying to feel for your texture, see, where are these Meissner's corpuscles located? Meissner's corpuscles, they are present in, they are numerous in glabrous skin. They are present in glabrous skin. Now, what is glabrous skin? What is glabrous skin? Glabrous skin is non-hairy skin. Non-hairy skin. So they are they are present in glabrous skin, especially fingertips. Fingertips, isn't it? Fingertips have a very high concentration of Meissner's corpuscles. Whenever you want to feel the texture, you use your fingertips, isn't it? So they're especially present in the fingertips. They are present on lips. They are present on nipples, right? So these are present on non-hairy skin, glabrous skin, especially fingertips, lips, nipples. Remember, these are Meissner's corpuscles. They are used for texture. They are used for topognosis. Topognosis. What is topognosis? Topo is topography. Gnosis is detection. Whenever you want to feel a surface, any kind of surface, if you want to feel it, you use your fingertips. Yes, and that is called topognosis. These questions have been asked for texture, for topognosis, and even in Braille. Even in Braille. What is Braille? Braille is a script for the visually challenged. There are elevations and depressions which are present on a piece of paper, and that is what is meant. They use their fingertips to read. That is called Braille. And Meissner's corpuscles, which are located in the fingertips, help these people to read. Right? So this is Braille, Meissner's corpuscle. So we've done two types of rapidly adapting receptors: Pacinian corpuscle, which is meant for fast vibration up to 800 cycles per second. And number two, we've done Meissner's corpuscle, which is specific for fine, well-localized touch, for slow vibration, for texture, for topognosis, and for Braille. There is another type of a rapidly adapting receptor. Let's see this. Rapidly adapting receptors are what are known as hair end organs. Hair end organs, another type of rapidly adapting receptors which are associated with hair follicles. Hair end organs, and hair end organs detect light movement on the skin, right? Hair end organs will detect light movement on the skin. Hair end organs will detect light movement on the skin. So these are three types of rapidly adapting receptors: Pacinian corpuscles, Meissner's corpuscles, and hair end organs. Okay? All right. Okay. This is as far as your the rapidly adapting receptors are concerned. Pacinian corpuscle, the Meissner's corpuscle, which is also an encapsulated nerve ending. It is encapsulated A beta fiber. Meissner's corpuscle is specific for fine, well-localized touch, slow vibration, texture, topognosis, and Braille and hair end organs. Let me complete this topic, receptors, and then we'll take a break. So I'll take another 5 minutes to complete it, and then we will do this. All right. Now the next is tonic receptors. Let's see tonic receptors. Now, as far as tonic receptors are concerned, tonic receptors, I told you, are slowly adapting receptors. Tonic receptors. Let's see which are the tonic receptors. The first one is Merkel's discs. Merkel's discs. Now, Merkel's discs are Merkel's discs are also, they have similar functions as Meissner's corpuscles. So Merkel's disc is present Merkel's disc, these are meant for fine, well-localized touch. This is meant for fine and well-localized touch. These are also meant for texture and topognosis. These are also meant for texture and topognosis, but if you're asked a question, which is a better receptor for texture and topognosis, your answer should be Meissner's, not Merkel's. Merkel's is also the nerve fiber which is associated as A beta, but if you have to choose between Meissner's and Merkel's for texture or topognosis, the better answer is Meissner's. Okay? Merkel's discs, they are present on both hairy and glabrous skin. They are present on both hairy and as well as the glabrous skin. Right? Both hairy and glabrous skin, Merkel's discs. Then you have another one which is called Ruffini's endings. Ruffini's endings. Ruffini's endings, again, the nerve fiber which is associated is A beta. Nerve fiber is A beta. Ruffini's is also meant for fine, well-localized touch. Fine, well-localized touch, meaning that tonic receptor means slowly adapting receptors. Please see your notes, you've written it already. Phasic receptors are rapidly adapting, tonic is slowly adapting. Topognosis. Topo means topography, gnosis is detection. Feeling a surface, whether it is rough, whether it is smooth, that is called topognosis. Okay? Ruffini's endings are fine, well-localized touch, and these are also meant for prolonged pressure. These are also meant for prolonged pressure. Next, C mechanoreceptors. C mechanoreceptors. C mechanoreceptors. These are meant for crude touch. C mechanoreceptors. These are meant for crude touch. These are meant for pressure. C mechanoreceptors, the nerve fiber associated is C. These are meant for crude touch and for pressure. Crude touch and pressure. Then you have the proprioceptors. Crude touch and pressure. Proprioceptors. Prasad, there is no specific location. The Ruffini's are present all over the hairy skin, all over hairy skin, right? Proprioceptors. Now, proprioceptors are, which are the proprioceptors? That means for joint and position cells. Which are the proprioceptors? Proprioceptors, you've got muscle spindles. Muscle spindles. Golgi tendon organs. Golgi tendon organs. Joint capsule receptors. Joint capsule receptors. Joint capsule receptor. These are meant for position sense. Position sense is joint. These are joint capsule receptors are different from Pacinian corpuscles. Pacinian corpuscles are located in joint capsules, but Pacinian corpuscles are meant for vibration. Joint capsule receptors are meant for proprioception. This is meant for position sense. These are your proprioceptors. These are also slowly adapting receptors for position sense, right? Muscle spindles, Golgi tendon organs, joint capsule receptors. These are meant for position sense. Then another one which is a slowly adapting receptor are the nociceptors. Nociceptors means these are free nerve endings for pain. What are nociceptors? These are free nerve endings for pain. Free nerve endings for pain. These are slowly adapting receptors, which makes so much of sense, isn't it? Let's say you've touched something hot. The moment you touch something hot, there is a pain that you feel which makes you withdraw your hand, isn't it? Right? So nociceptors are slowly adapting receptors. Supposing my nociceptor was a rapidly adapting receptor, what would have happened? You've touched something hot, your hand has got burned. If my pain receptor was rapidly adapting, the pain sensation would have stopped. I would have forgotten that I have injured my hand, there is a burn. I would have probably gone and touched again, producing even more tissue damage. Have you understood? So nociceptors are very poorly adapting receptors. Pain, very poorly adapting receptors. You continue whenever there is an injury, very poorly adapting receptors, or sometimes not at all, right? Or sometimes not at all, which makes so much of sense because pain is protective. Whenever there is an injury, whenever let's say you are running, you fall down when there is an injury, because of the pain, you stop that activity. You protect yourself against further tissue damage. So nociceptors are free nerve endings for pain, which are very poorly adapting receptors or sometimes not at all. And what are the types of nociceptors? Nociceptors can be of 4 types. These are, let's say, mechanical nociceptors to mechanical pain. Yes, pain of a pinprick or somebody gives you a pinch, that is mechanical. There could be thermal nociceptors, which respond to changes in temperature. There could be chemical nociceptors, which respond to inflammatory chemicals. They could be polymodal nociceptors. What are polymodal? They respond to more than one type of pain. This is what is known as nociceptors. Nociceptors are very poorly adapting receptors or sometimes not at all. Have you understood? So these are your somatic receptors. Yes, which are the somatic receptors? Let's have a look. Slowly adapting receptors, Merkel's discs, which are located on hairy, glabrous skin, fine, well-localized touch, also for texture and for tropognosis. If you are asked for fine, well-localized touch, which is a better receptor, your answer should be Meissner's. For texture, Meissner's. Tropognosis, Meissner's. Braille, Meissner's. Yes? Ruffini's endings. Fine, well-localized touch and for prolonged pressure. C mechanoreceptors, crude touch, pressure. Proprioceptors, position sense, muscle spindles, Golgi tendon organs, joint capsule receptors. Nociceptors, which are free nerve endings for pain. They are very poorly adapting or sometimes not at all. Right? Another one, which is a poorly adapting receptors, are the thermoreceptors. Thermoreceptors are also slowly adapting receptors. Thermoreceptors, these are slowly adapting receptors. They will detect temperature, right? Temperature. Now, thermoreceptors, they can be receptors for cold and receptors for warmth, right? Polymodal means they respond to more than one respond. Poly, more, modal, modes. So they respond to more than one type of pain, more than one type of pain. That means they can respond to mechanical, respond to thermal, they can respond to chemical. Polymodal, more than one type. Tonic is another name for slowly adapting. This has already been done. All right. Now, thermoreceptors, they can be cold thermoreceptors or they can be warm thermoreceptors. Warm thermoreceptors. Cold thermoreceptors, these are more numerous. These are more numerous. There are more cold sensitive spots. These are more cold sensitive. There are more cold sensitive spots on the skin. Warm thermoreceptors are less in number. Cold sensation is carried by the A, delta, and C fibers. Warm is only by C, which we've already discussed. Warm can detect temperature. This can detect temperatures between 30 and 45. Ojas Bano, thermoreceptors will detect temperature. The thermal nociceptors, when the temperature is excessive, when you touch something hot, that is a thermal nociceptor, painful, when temperature becomes painful, right? Okay. So please listen to this. Let me just complete this and then we'll take a break. Just this last topic, thermoreceptors. Yes, they can detect are Santosh, where are Golgi tendon organs? In the tendons. It is given in the name. Warm thermoreceptors can detect temperatures between 30 and 45 degrees Celsius. Okay, 30 and 45 degrees Celsius. Now, whereas the cold thermoreceptors will they are activated at temperatures. At a temperature, the activation begins at temperatures. Activation begins at a temperature of 40 degrees Celsius. Please understand, these thermoreceptors are not meant for temperature regulation. They are meant for detection of temperature on the skin. Have you understood? For example, if I place an object, right, like this is a glass of cold water, if I place it on the skin, this is going to activate my thermoreceptors. Have you understood? This is what I meant by thermoreceptors. This is what is meant by somatic receptors. Some people saying, what is somatic? Receptors present on the soma, on the periphery. They are different from visceral receptors, which are present inside, right? Or interoceptors, which are present inside the body, right? So these are somatic receptors. They are meant for detection of temperature on the skin. Cold thermoreceptors begin to be activated at temperatures of 40. Now, cold thermoreceptors, 40 degrees Celsius. Bharat Kumar Soni, the nerve fiber which is associated with warm thermoreceptor is C. We have already done that. Yes. Now, at 40 degrees Celsius, they begin to get activated, the cold thermoreceptors. The activation increases as the temperature goes below 40. Maximum activation of these receptors will be at a temperature of 24 degrees Celsius. When you place an object at 24 degrees Celsius on the skin, maximum activation of the cold thermoreceptors, then the activation of the cold thermoreceptors reduces, and less than 10 they stop responding. This is what is the unique activity of the cold thermoreceptors. The activation of the cold thermoreceptor will begin if I place an object at 40 degrees Celsius on the skin. If an object is at 24 degrees Celsius, maximum activation of these cold thermoreceptors. These are somatic receptors. They are detecting temperature of the objects placed on the skin. If an object at 24 degrees Celsius is placed on the skin, maximum activation of the cold thermoreceptor. As the temperature of the object goes below 24, the activation of cold thermoreceptor reduces. Less than 10, they stop responding. So that means if you have, supposing I ask you a question, this is a bowl of water which is at 5 degrees Celsius. If I place your hand in this bowl of water, which receptors are getting activated? Is it the cold thermoreceptor or is it the nociceptor? Bolo. Is it the cold thermoreceptor or the nociceptor? If I place it at 5 degrees Celsius, this will now there will be a nociceptor activation. It is not the cold receptor activation. Which nociceptor? The thermal nociceptor, which responds to temperature. Have you understood? This is not going to activate the cold thermoreceptor because cold thermoreceptor stops. Activation of cold thermoreceptor stops at temperatures less than 10. Okay? Now, another important point is for objects placed on the skin, for objects placed on the skin and between 30 and 40 degrees Celsius, for objects placed on the skin between and at and at temperatures between, placed on the skin and at temperatures between 30 and 40 degrees Celsius, both cold and warm thermoreceptors will be activated. There is an overlap, isn't it? Both cold and warm thermoreceptor activation will be present for objects placed on the skin and at temperatures between 30 and 40 degrees Celsius. Both cold and warm thermoreceptors will be activated. But if the temperature of the object, so between 30 and 40, I said there is an overlap. Yes, there is an overlap. But if the temperature of the object is at 30, cold receptor activation is plus. If the temperature of the object is at 40, warm receptor activation is plus. I've understood. So this is what is the thermoreceptors. Thermoreceptors are also slowly adapting receptors. Yes. Now, if you look at these thermoreceptors, you've got two types of thermoreceptors: receptors for warmth, receptors for cold. Which are more numerous? Cold. There are more cold sensitive spots on the skin as compared to warm. Cold sensation is carried by A delta fibers and by C fibers. Warm sensation is carried only by C fibers. We've discussed that. Now, what is the temperature? Warm thermoreceptors can detect temperatures between 30 and 40. These are temperatures of objects placed on the skin. Okay? 30 and 45 degrees Celsius. Cold thermoreceptors, they begin to get activated at 40. Maximum activation at 24. The activation reduces. Less than 10, they stop responding. If I place your hand in cold water at 5 degrees Celsius, you are activating the thermal nociceptors, not the cold receptors. Between objects with temperatures between 30 and 40, both cold and warm thermoreceptor activation will be present. But if it is the temperature of the object is at 30, cold receptor activation is plus. If it is at 40, warm receptor activation will be plus. Okay? All right. Okay. Now, this is as far as your different types of warm and cold thermoreceptors are concerned. Different types of phasic and tonic. So let me just tell you once again, which are the phasic receptors? Phasic receptors are the rapidly adapting receptors, encapsulated nerve endings. And what are the examples of phasic receptors? Number one is Pacinium corpuscle, which is a very rapidly adapting touch receptor, but it is not a receptor for touch. It is specific for fast vibration up to 800 cycles per second and for deep pressure. Where are the Pacinium corpuscles located? Joint capsules and deep in muscles and fasciae. Meissner's corpuscles, fine, well-localized touch, slow vibration, and texture, topognosis, braille. So you've got, if you have a question for fine, well-localized touch, Meissner's; for texture, topognosis, and braille. Remember, braille, he is running his fingertips rapidly on a paper which has got elevations and depressions, right? So this is your for hair end organs, which are associated with hair follicles, and they detect light movement on the skin. Tonic receptors, slowly adapting receptors, Merkel's discs, Ruffini's endings, C mechanoreceptors, proprioceptors, nociceptors of 4 different types: mechanical, thermal, chemical, polymorpal, and the thermoreceptors. Okay. Cold thermoreceptors begin to get activated at 40. The activation increases at 24, decreases to 10. Okay? So that means they're getting activated between 40 and 10. Okay? All right. See, nociceptor will be whenever there is some sort of tissue damage. So more than 45 or less than 10. Okay, chalo. Take a lunch break. Yeah.